And all right, we're good. We are good now. Thanks for your patience. Let's go ahead and start. So this exam covers chapters 12, 14, 15, and I will go over some of the major things to understand to do well on the exam. So obviously know what true breeding means. What is true breeding? We talked a lot about Mendel. So his big contribution was the monohybrid cross and the dihybrid cross. You remember that? What do we need to know about both of these? First of all, you start with true breeders. Okay, so know what a true breeder means. Then we formed the hybrid, and the hybrid was self-fertilized to give us the F2 generation. So when on the test, when you get the question, what is the phenotypic ratio, or what is the genotypic ratio, it's talking about the F2 generation, okay, specifically. So what is the phenotypic ratio of, say, a monohybrid cross? What was that ratio? 3 to 1. Remember, 3 purples to 1 white flower, for example. In this example here, 3 to 1, okay? So what's the genotypic ratio? 1 to 2 to 1, right? One of them was a true breeder, homozygous dominant, which means big P. 2 of them were non-true breeders, so that's heterozygous, which means big P, little p. And one of them was homozygous recessive, which means little p. Remember that? So again, for exam reasons, the phenotypic ratio of a monohybrid cross is 3 to 1. The genotypic ratio of a monohybrid cross is 1 to 2 to 1. Okay. And what did we learn from the monohybrid cross? Let me skip ahead a couple of slides. What was the takeaway message Mendel gave us? The principle of segregation of alleles. Does that make sense? This is what we learned from the monohybrid cross, that each individual has 2 alleles for every 1 gene, for every 1 trait, and they segregate into the progeny, into the sperm and the egg, into the gametes, and then they come together upon fertilization. Does that make sense? So if you see what did we learn from the monohybrid cross? Well, Mendel showed us the principle of segregation of alleles. Okay. And what else did we learn from the monohybrid cross? We learned that there aren't intermediate traits, right? There's no blending. The alleles are discrete. What does that mean? There is no purpley white flower. It's either purple or white. Okay? That means that the alleles are discrete. Okay, that's an important thing he showed us. There was a dominant allele and a recessive allele, right? So we should know what that means. We should know what genes are. Genes, Mendel would have called this traits, right? And then alleles are alternative forms of a gene. So, for example, if we're talking about eye color, that's the gene. If we're talking about blue eye color, that's an allele. Okay, you see it's an alternate form of that gene. Homozygous means 2 of the same allele, like blue, blue, brown, brown, little b, big B, big B. You see what I'm saying? What would Mendel call homozygous? What would Mendel have considered those organisms? True breeders, right? Homozygous means you have big P, or little p. That's what homozygous means. To Mendel, he didn't know about alleles, so he would have called it true breeder, right? Heterozygous then means non-true breeder. It means you have 2 different alleles, a dominant allele and a recessive allele, like a big P and a little p, a big B and a little b, right? So hopefully pretty straightforward stuff. And then, and if you're studying these types of Mendelian traits, 1 allele is dominant, like purple, big P, and 1 allele is recessive, like white, little p. Make sense? Dominant allele, recessive allele. And then do you remember what genotype means? Genotype means what alleles do you possess? What alleles does this creature possess? So if it's a true breeder, its genotype is big P, or little p. Does that make sense? If it's a non-true breeder, its genotype is big P, little p. Okay? Makes sense? Phenotype, on the other hand, is what you see. Remember what you see. So is it purple or white? Punnett squares, you guys remember how to set these up? The very first thing you need to know is what is the genotype of the mom and what is the genotype of the dad, right? That's the first thing. The second thing you need to know is what kind of gametes can this parent make? What kind of gametes can that parent make, right? So if your genotype is big P, little p, what kind of gametes can you make? What could be in a sperm? Big P or little p. Can you have two P's in one sperm? No, because that would mean the sperm is diploid, and sperm are not diploid, they are haploid. And egg as well, egg's the same. So what you need to know for the exam reasons, for exam purposes, is you can't have two of the same allele, two of the same letter in a gamete, in a sperm or an egg. You cannot have two P's in one. You could be a diploid organism with a big P and a little p, but your sperm or egg have one or the other. They cannot have both. Okay, so you put the egg information on one side of the square and the sperm information on another side of the square. It doesn't matter which side you pick, and then you solve the square. Okay, and then we talked about the dihybrid cross. Okay, we talked about the dihybrid cross, where you're looking at two traits at the same time. Again, you're starting with true breeders, and then you made a hybrid, you self-fertilized the hybrid to get this, right? This is the F2 generation. So I might ask, what is the phenotype of the dihybrid cross? What is the phenotypic ratio? So what is that? 9, 3, 3, 1, right? 9 dominant dominants, 3 dominant recessives, 3 recessive dominants, and only 1 that's recessive for both traits, right? Now, would I be mean enough to ask what is the genotypic ratio of a dihybrid cross? No, because that would be pretty long, right? I mean, basically what that is asking is, you see how the different genotypes, big R, big R, big Y, big Y? Well, how many of those are there? And then big R, big R, big Y, little y's, well, how many of those are there? And by the time we get done, it's going to be a really long ratio. So I'm not saying I won't ask it on the test, okay? But I'm saying if I do ask it, unless you see an answer that's about this long, it's most likely what answer? None of the above, okay? No, it's probably none of the above. And you know, that's a hint. Anyway, for the genotypic ratio of a dihybrid cross. You should know the phenotypic ratio, but if you see the question genotypic ratio, you know, you pretty much know it's going to be a none of the above type of thing, right? And what did we learn from the dihybrid cross? What was the takeaway message that Mendel showed us? The principle of independent assortment, right? What does that mean? That different traits assort independently of one another. And now we know why, because of metaphase 1. When independent assortment happens, those tetrads line up randomly, right? So good. And a test cross. This is an experiment we could do. When do you conduct a test cross? You should know for the exam, when do we conduct a test cross? When you want to figure out what? When you want to figure out the genotype of an organism with a dominant phenotype. Does that make sense? So I want to figure out if this purple flower plant, for example, is big P or big P little p. I can't tell just by looking at it, can I? No. That's why I do a test cross. So a test cross is done to figure out the genotype of an organism with a dominant phenotype. Would you have to do a test cross to figure out the genotype of a recessive phenotype? No, if I gave you a white flower plant and you knew white flowers were the recessive color, you already know the genotype. It has to be little p, it has to be, yeah. You wouldn't have to do a test cross. And what is a test cross? You just cross it with a recessive plant, and based on the results, you know that you've answered that question, haven't you? If it was a true breeder, if it was big P, or non-true breeder, big P, little p. Any questions about chapter 12? If you followed along, you're nodding, you're understanding, this is what we're needing you to know, and you're going to do just fine on the test. Any questions about that? By the way, again, there might be questions like, this organism with this genotype can make all of the following gametes except And what kinds of things are you looking for? You're looking for, for example, can you have a R and a Y in a gamete? Yeah, you can have an R and a Y in a gamete because R has to do with shape and Y has to do with color, right? But can you have two R's in a gamete? No. Can you have two Y's in a gamete? No. Those are the types of reasoning you need to be able to understand for the test, right? Okay, any questions? Chapter 12, pretty straightforward. All right, good. Okay, let's go on to the next chapter. Chapter 14, I also want to see the battery status. Seems okay. Good. All right, guys, let's go on to 14. All right, what do we need to know about 14? A lot of the beginning of this is review, so I'm not going to spend a ton of time on it. Just remind yourself, what are nucleotides? Remember, they have a phosphate group, a nitrogenous base, a 5-carbon sugar called deoxyribose for DNA. On the 5' you have a phosphate group. On the 3' you have a hydroxyl. Do you remember the purines versus the pyrimidines? Remember that mnemonic device I gave you? One pyrimid could cut you. One meaning the one-ring nitrogenous bases are the pyrimidines, and they include C's, U's, and T's. I may or may not ask about that, but that's good to know. In DNA, you have thymine. In RNA, thymine's counterpart is uracil, right? So RNA uses U's, whereas DNA uses T's. You link nucleotides together by phosphodiester bonds. And when you do that, you end up with a linear molecule. DNA and RNA are linear. They have a 5' end with a phosphate group and a 3' end with a hydroxyl. We should know that it was mainly Rosalind Franklin's work and her boss Maurice Wilkins' work that allowed us to understand the structure of DNA. It was not Francis Crick and James Watson. Okay, what else? We should know that the two strands of DNA are antiparallel. We should know that G's complement C's with 3 hydrogen bonds, and A's complement T's with 2. All of this is a review for the most part. Oh, I might ask, in what fashion is DNA replicated? And which one's correct here? Semi-conservative. Yeah, so you should know DNA is replicated in semi-conservative fashion, right? That's the correct way. Which way is DNA synthesized? In what direction? 5 to 3, 5' to 3', with respect to the new strand, the daughter strand. Good. And the enzyme that does most of that copying is called DNA polymerase III in prokaryotes. And do you remember that DNA polymerase III can't copy DNA from scratch? It can't start a second strand. So the first thing you have to do is put down a primer. The primer is the short stretch of RNA, and primase, or RNA polymerase, is the enzyme that puts down the primer. Then it detaches, and what attaches? DNA polymerase III to carry on the rest of the copy. Good, and that all is happening in a 5' to 3' direction, remember? Real quick, prokaryotes, remember how they have one circularized chromosome? How many origin of replication do they have? 1, that's what you need to know. Prokaryotes have 1 origin of replication. Eukaryotes, they have a lot more to copy, so they have multiple origin of replication. That's what you need to understand for the exam. On this table, what you need to know about is DNA polymerase I and DNA polymerase III. You don't need to know about 2. DNA polymerase III is the enzyme that it copies DNA going to 5' to 3, and it copies most of the DNA, right? DNA polymerase I, you should know, that's the one that comes in afterwards, removes the RNA primers, replaces it with DNA, right? We should know that. We should know that helicase is the enzyme that unwinds or unzips the DNA. We should know that single-strand binding proteins keep the 2 strands apart. And we should know that topoisomerase, aka gyrase. Okay, gyrase, topoisomerase, same thing. Gyrase is an example of topoisomerase. This one corrects overwinding ahead of the replication fork by breaking, allowing the DNA to unswivel and reannealing the DNA. So it basically corrects the overwinding. It corrects that supercoiling we talked about. Then we talked about leading strand and lagging strand. Remember the lagging strand, you get Okazaki fragments because you keep building backwards. You need multiple primers. What do the beta clamps do? Remember the beta clamps? Processivity. For exam purposes, you need to know the beta clamps increase the processivity of DNA polymerase 3. What does that mean? It's a fancy way of saying keeps DNA polymerase 3 on the DNA doing its job. Makes sense? And do you remember what each of these players do? Primase puts down a primer. DNA polymerase 3 copies the DNA. DNA polymerase 1 replaces the primers with DNA, but leaves a little break in the backbone, right, between the fragments. And what fuses the fragments together? DNA ligase. You should know DNA ligase joins the Okazaki fragments. And do you remember the clamp loader? The clamp loader's job is to load the clamps. Okay, and you remember what the beta clamps do? Increase the processivity of DNA polymerase 3. Now you don't need to go into these slides here and know exactly what the clamp loader's doing and exactly step by step everything. I have it here for your own information because it's neat to understand, but you don't need to stress out about this. Just know what the players do, right? Do you remember in eukaryotes, again, you have a lot more DNA and you have linear DNA instead of circularized DNA, right? So because of that, you have multiple origin of replication instead of just one origin of replication. And you also have the ends of the chromosomes to deal with, right? The ends of the chromosomes have a name. What was the end of our chromosome called? Telomeres. The ends of our chromosomes are called telomeres. And do you remember there's a problem with our telomeres? The last primer, when you remove that last primer, it's hard to fill in that DNA because you need something to build off of, right? And what solves that problem and prevents shortening of our telomeres? Telomerase. That's what you need to understand. We have an enzyme called telomerase, which prevents the shortening of our telomeres by filling in those telomeres, something that DNA polymerase 1 can't do. Okay. That's what we need to know. Telomerase prevents shortening of telomeres by filling in the final primer with DNA. And that's it. That's this whole chapter. If you understood those things, those concepts, you should do just fine. Any questions you guys have? No, pretty straightforward. And this last exam is straightforward. You know, you just learn the material, get it clear in your mind, straightforward, you'll do fine, I'm sure. That's it for this chapter. Let's go on to the final chapter. And let me check the battery status. My phone is on lower battery today. We're good. Okay. Let's move on to the final chapter, chapter 15, and see what we need to know here. This is an exciting chapter, gene expression. So you should know the first part of gene expression is transcription, where you copy the gene to messenger mRNA. Or that's redundant, isn't it? mRNA means messenger RNA. And then you have translation, which is using the mRNA, the ribosomes can read that message on mRNA to produce protein, right? Transcription, translation. So do know about those terms. Now, this is important to understand for the exam, the difference between prokaryotes and eukaryotes with gene expression. In prokaryotes, see the picture on the right, there's no nucleus. So transcription and translation happen in the same place. That's what you need to know. And that place is the cytoplasm. And they happen at the same time. What do I mean? Before transcription's even done, you can start translation. Does that make sense? You know why? Because you see the mRNA, it's matured the minute it's made. It doesn't need to be processed. Is that clear? And in the eukaryote, see on the left, or my left, your right, the eukaryotic cell has a nucleus, doesn't it? And the DNA is inside of there. So transcription has to happen in a nucleus. Translation happens in the cytoplasm, right? And so it's separated by space and time. Transcription happens first in the nucleus. Translation happens second in the cytoplasm. And not only that, look, you have an extra step between the two. You have RNA processing. Remember those three modifications? 5' cap, 3' poly-A tail, and excision of the introns, removal of the intron information. Remember that? That only happens in eukaryotes. You should know what codons are, right? Blocks of 3 nucleotide. Now you don't need to memorize this entire table because that would just be me, okay? What you do need to do, though, you need to memorize 4 of these. You need to know the start codon is AUG. On the DNA, it would be ATG, right? AUG, and you should know the stop codons. UAA, UGA, UAG. So do know the stop codons and do know the start codon. And what amino acid does the start codon also code for? Methionine. See, the start codon AUG, not only does it mean start, it means methionine, the amino acid methionine. Fun fact, all proteins, because of that, start with methionine, right? That's an interesting thing to understand. Okay. You should know that the genetic code is degenerate or redundant. That means multiple codon can code for the same amino acid. But you should know that the genetic code is not ambiguous, which means that a particular codon will never code for more than that particular amino acid. Okay, so do know that statement at the top for the exam. You should know that the genetic code is practically universal. With respect to, you see this whole transcription initiation, you don't need to know it in great detail. What I really need you to know is what a promoter is. You remember what a promoter is? Look, the promoter is a stretch of DNA upstream of the gene. That means in front of the gene, where the enzyme RNA polymerase binds in order to start transcription, right? Does that make sense? So the promoter is just a stretch of DNA in front of the gene. That's where RNA polymerase binds so that it can go and copy the gene. Makes sense, right? That's what we need to understand. And mRNA is synthesized in what direction? 5 to 3. Just like DNA is synthesized 5 to 3, RNA is synthesized 5 to 3. Those are the things to understand. And by the way, another important thing to understand for the test, in prokaryotes, the RNA polymerase 3 attaches directly to the promoter, right? It doesn't need transcription factors, does it? But what about in eukaryotes? Eukaryotes, yes. Eukaryotes require transcription factors first, then RNA polymerase binds. So prokaryotes don't require transcription factors. Eukaryotes, they do require transcription factors to start transcription. Right? Make sense? That's the main thing to understand. What else? Yeah, keep going. You don't need to know the difference between polymerase 1, 2, 3. You don't need to worry about that. Termination, you don't need to worry about that. Now, only in eukaryotes you have this, right? It's called DNA modifications or mRNA modification or mRNA processing, right? You've got 3 modifications you need to do, right? Remember, 5' cap, 3' poly-A tail, and removal of what part? The introns, removal of introns. A fancy way of saying removing introns is excising the introns. Excising means removing the introns. Keep that clear for the exam. What are you excising? The introns. I mean, it sounds nicer to say you're excising the exons. That actually sounds, but that's not. You're excising the introns, okay? Those are the 3 modifications. And you don't need to know it in great detail. You don't need to know it's methyl, guanine, 5' to 5'. You don't need to go into that kind of detail. Just know the modifications. 5' cap, 3' poly A tail, excise the introns. Right? Does that make sense? Very straightforward. And the enzyme that does the excision of the introns is called splicing out the introns. The spliceosome. The spliceosome is the enzyme that does the splicing out of the introns. It does the excision of the introns. This is an interesting thing too. Remember alternative splicing? You should know for the exam, this is responsible for the fact that we have more proteins. We have like 80,000 different proteins when we only have 25,000 different genes. Because remember, the math doesn't add up. If you have a gene, it codes for a protein. If you have 25,000 genes, you should have 25,000 different proteins. But we have like 80,000 different proteins, so the math doesn't add up. Well, what's that discrepancy? Alternative splicing, right? If you splice out, for example, alternative splicing, one way of splicing this is putting exon 1, 2, 3, and 4 right next to one another, right? But an alternative to that would be, what if I left out exon 3 completely? What if I just left exon 3 out as well? What if I splice that out? Well, then I would have a transcript, a mature mRNA that has exon 1, 2, and 4. You see? That's an alternative way of splicing, isn't it? That would give me a different mRNA, and if it gives me a different mRNA, it's going to give me a different protein. So that's 2 different proteins right there that I could make from the same gene. Does that make sense? So alternative splicing is something that allows you to have more variety of protein than you have variety of genes. All right, does that make sense? Can prokaryotes do alternative splicing? No, because they don't do splicing, period. Right, does that make sense? Good. Now, what's something that prokaryotes can do that eukaryotes can't, right? Do you remember this concept here? Trying to go back. Operon. Do you guys remember the concept of operon? So operon means one promoter is being shared. Just one promoter for more than one gene. That's weird, right? And usually those genes are functionally related. Okay? So you find these in prokaryotes. Prokaryotes have operon, which means they can have one promoter for multiple genes. This allows you to quickly, more rapidly produce those proteins, right? And eukaryotes just don't have operon. Eukaryotes have one promoter per gene. That's what you need to know. So prokaryotes have operon. Eukaryotes have alternative splicing. Okay, makes sense? What else? Good. We talked about tRNA. Here's what you really need to understand. With the translation, what is the P site doing? What is the A site doing? What is the E site doing? Okay, so let's start with the P site. It's where the peptide bond is made. What is the proper term for that? I'm trying to find it. Peptidotransferase activity. Remember that? I'll find it here. Peptidotransferase. So what you need to know for the exam is that the P site is responsible for peptidotransferase activity, meaning makes a peptide bond. Right? Remember, it links the amino acids to one another. Yeah, it makes the peptide bond. Okay. What does the E site do? Eject, right? And what does the A site do? Accepts the charged tRNA. Okay, so that's what we need to know for the exam. A site accepts charged tRNA, E site ejects tRNA, and the P site, peptidotransferase activity, makes the peptide bond. Very straightforward stuff. Remember, translation starts at the 5' end, right? Translation starts at the 5' end of the mRNA. And then you look for the AUG, and you start translation, right? You don't need to know everything in great detail here. Just the main concepts here. What else do we need to know? We already covered this. Again, you don't need to know the elongation cycle in great detail, step by step. Don't worry about it. Know the main concept here, right? Again, A site accepts charged tRNA, P site makes the peptide bond, and then E site ejects. Very straightforward. You don't need to know what wobble is. You don't need to know it. Now, here's something you do need to know. When you encounter a stop codon in the A site, is there a tRNA that recognizes the stop codon? No. No. That's what you need to know. It is a protein that recognizes the stop codon. That protein is called release factor. Okay, does that make sense? Good. Good, good. And then this is all that's left. Types of mutations. What kind of mutation changes the DNA but not the protein? Not the amino acid, not the protein? Silent. What kind changes the DNA and the amino acid? Missense. And what kind introduces a premature stop codon? Nonsense, right? You don't need transversions and transitions. You don't need to know that. You don't need to know that. And here's something you do need to know, and this is, okay, this is going to be like a wink-wink moment, right? As maybe a, bonus-y type extra-y thing, right? That wasn't a very subtle hint. And that is that sickle cell is an example of a missense mutation, right? Sickle cell is an example of a missense mutation. Why? Because GAG was mutated to GTG. It's just an A to T mutation. But now, instead of having a glutamine in this hemoglobin, you have a valine on this position. So you've gone from a polar glutamine amino acid to a non-polar valine, and that caused the protein to not fold correctly. It didn't fold correctly. It has this exposed hydrophobic region that causes them all to concatenarize, which means clump up. And that stretches the cell out. You see, that simple one little missense mutation results in a lifelong disorder. That's what genetic disorders are. Okay, so be able to explain that maybe. That would be nice to understand. And then remember, frame shift, this is caused by either inserting or deleting a nucleotide. Okay, and that shifts the reading frame. Okay. Any questions you guys have? Let me go ahead and stop the stream now because that is the end of the review.